Alright, I'm back, and I want to note that although I am not wearing gloves, it's because I have to work my iPhone, and plus I'm old and I already had my kids and I have no retirement plan, so I have to die before I reach 70. So I would recommend absolutely wearing gloves. It does take a little bit to get that smell out of your hand. I had to wash my hand 3 times because I didn't wear these gloves right here. Alright, so the first thing we're going to do is remove the dura mater covering, right, this sheath of connective tissue around there that protects the brain. Right, and we're going to remove that, and one thing to keep in mind here is the ventral structures, or those structures down over here, are going to get removed, right? So we're going to point out some things before we do remove those. So I'm going to start on the anterior side, right here near the olfactory bulbs, and cut a strip down the dura mater, right, down that longitudinal fissure, right, to separate those two halves first, right? And one thing you might want to do is take a probe and kind of separate in between the brain and the dura mater to make it easier to make that cut. Right, so then you're going to cut down that fold just on one side of the longitudinal fissure, and then cut all the way back till you reach around the cerebellum. And then I'm going to go back around the front there and snip away any remaining attachments on the anterior side and on the ventral side. And then note here, I'm going to cut around as best as possible the optic nerves right there so that they remain intact right there. So I'm going to remove the cerebellum now from the ventral portion of that, avoiding cutting the optic nerve as well as those other structures in there, which we'll leave until the end. So I'm going to keep on cutting posterior here till I reach that place where I stopped on the longitudinal fissure around the cerebellum. And then we could remove that whole side of the dura mater. And because I cut it a little off to the side right there, we're still going to be able to make out the dural sinus, which is within the dura mater, right over there. And we could also see underneath it, we could see that falx cerebri. Right, that's going to separate the left and right hemisphere. All right, then the next thing we're going to do is remove the last of that dura mater that's covering the cerebellum. Right, it's a little bit hard right there because they're going to have that tentorium cerebellum within the transverse fissure. Right, and the transverse fissure, shown right here, separates the cerebellum on the posterior side with the cerebrum on the anterior side. Alright, so now most of the dura mater is removed, with the exception of some of the ventral stuff that's covering there. But before we leave the dura mater, just get a feel for the texture of that dura mater. You could also see the dural sinuses, that is that space in between where those vessels are going to run, right, to drain the cerebral spinal fluid. And you could also see on the posterior side that tentorium cerebelli, right there, that was separating the cerebrum from the cerebelli in the transverse fissure. All right, so once we've removed the dura mater, we could also check out the arachnoid mater, right, and get a feel for the texture there. Remember, arachnoid means some kind of spider reference, and you can see how it looks kind of like a spider web, right? So underneath the arachnoid mater is the subarachnoid space, where there would be cerebral spinal fluid, right? And so that has been drained during the fixation process, right? But underneath this space right here would be the cerebral spinal fluid. You can't really see the pia mater at all, but that's the arachnoid mater. And so last, we're going to remove this last ventral piece of dura mater that covers the pituitary gland and some other structures. But in the meanwhile, we're going to try to keep the optic nerves intact while doing so. Right, so that's cranial nerve number 1, and also underneath this dura mater, coming off the pons, you're going to see these two paired nerves. These are the oculomotor nerves, left and right ones that are going to be going through the dura mater, the cranial nerve number 3, the oculomotor neurons. And they would be passing through the dura mater and going over to innervate the muscles of the eye. But we're going to try to keep that intact as we remove that last part of the dura mater there. So as you lift up the dura mater, you could just snip off the oculomotor nerves at the distal end, and then they'll be there for a marker of the cranial nerves that are coming right off the pons right there. Right? So there's cranial nerve number 3, oculomotor. And then right underneath it, again, is the pons, right? That region that kind of bulges out, right? So that means the cranial nerves are coming off the pons and they're running over the midbrain region right over there. All right, so back to the superior view. And you want to orient yourself by finding those olfactory bulbs, and then you'll know you're in the anterior position, which remember is the location of the frontal lobe, whereas the cerebellum over there will mark the easier kind of posterior section right there, and then the back of the cerebrum would be the occipital lobe. All right, so here's the ventral side again. There are those cranial nerve number 3, the oculomotor neurons, right, innervating your eye. So remember your eyeballs, or the sheep's eyeballs, sitting in the front of the head. Their sensory information, the photoreception, is coming from the optic nerves over there, whereas these oculomotor neurons are going to be innervating the muscles of the sheep's eyes. So those oculomotor neurons number 3 are good to associate with coming off the top or the anterior portion in the sheep, or superior portion in the humans of the pons. And then right superior or anterior to the pons is the midbrain, right? And here we're looking at the anterior surface, again, of the midbrain, but the more distinguishing aspect of the midbrain will be on the posterior surface. There you have those structures known as the paired superior colliculi that are on the top that mediate visual reflexes, and on the inferior side you have the inferior colliculi which mediate auditory reflexes. You'll notice that these are much bigger relative to the cerebral cortex than they are in the human. And so together the paired superior and inferior colliculi make up the corpora quadrigemina, right? That structure on the dorsal midbrain or the posterior midbrain. So those structures are good kind of landmarks to find what's on the anterior surface if you follow them around, right? So here are those superior and inferior colliculi on the back, and if you follow them around to the front, right there, well, that'll be the anterior midbrain. And then just below that will be the pons, just inferior to the midbrain. And then below the pons, we'll have the medulla oblongata, which will lead into the spinal cord. And of course, that big lump behind the pons and medulla is the cerebellum. In this case, you can't really see the folia of it, the folds, because this one is kind of a mushy preparation. All right, so now we're back looking at the superior surface, and we're going to begin with this sagittal cut. But before we do that, we want to cut down the longitudinal fissure very carefully, because we want to expose the corpus callosum that is going to be running, that white matter tract that's going to be running from the right to the left side of the brain. And so that connects the 2 cerebral hemispheres, and notice here it does have that white appearance, which won't be so obvious when we look at it from the sagittal view. So now we're going to continue that cut down the longitudinal fissure and trying to get as close as possible to the midline when we go through the cerebellum and the brainstem structures. Here we're looking at the sagittal view now, and the preservation of this sample isn't that great, but we can make out the major brain structures here, such as the cerebrum. And within the cerebrum, remember, we have that white matter tract, the corpus callosum. That will be obvious, especially when we expose the ventricles. And the oval-shaped structure, some cases it'll be more obvious than others, is the thalamus, sitting below the corpus callosum. And then underneath the thalamus, we're going to have the hypothalamus. Right? So thalamus and hypothalamus make up the diencephalon. The next region underneath the thalamus and hypothalamus is the midbrain structures, or the mesencephalon. Inferior to that comes the pons, and then inferior to that is the medulla oblongata, right? And that will lead into the spinal cord going down the vertebral canal, right? And of course, the cerebellum is the biggest lump on the back of there. So this is it from the sagittal view. And the last thing I'll point out, on the cerebellum here, you could see the white matter tracts going into the cerebellum, right? And those are the arbor vitae, otherwise known as the tree of life, for the branching pattern that we see here of the cerebellum. All right, so back to the sagittal section of the big brain area. Let's look at the structures like the lateral ventricle in the middle when I pull it open right there. And then right above that lateral ventricle, we're going to see the corpus callosum. And then right below the lateral ventricles will be the other white matter tract called the fornix. So you'll relate those to the structures that are going to be inferior to that. So there's the corpus callosum, and then the lateral ventricles around the other side, and then the fornix, and then right below that, or inferior to the fornix, will be the thalamus. And then just under the thalamus there, of course, will be the hypothalamus. And then at the end of the epithalamus, which you can't see here, is the prominent pineal gland, again, which secretes melatonin, because even sheep need something to sleep. So they're all structures of the diencephalon, and then the next region will be the mesencephalon, or the midbrain area. So those are some of the regions and structures that are visible on this sagittal point of view. So you want to relate them to each other as well as to the human brain and look at the similarities and differences. And so that's it for the sagittal view. I also want to look at a coronal view, and what you want to do here is try to get a line roughly about in the middle of the thalamus, and then you could just cut that section here to take a look at the gray and white matter arrangement. Right, so here is the thalamus, right, which you could see within. That'll be the third ventricle around it right there, and then the cerebrum over here, right? So here is the fornix, and over here is the corpus callosum. And you can see from the coronal view on the right side here how it actually takes on that more white matter appearance. So from this coronal view, let's take a look at that gray and white matter, and in particular these gyri, which, although they're not nearly as prominent as they are in humans, are evident from this perspective. And so here's a prominent sulcus, that is that groove that's going in between the gyri. And last, we'll try to imagine what's going on microscopically. The neuron cell bodies within the gray matter extend their axons out through the white matter to connect with other neurons, going down to maybe the spinal cord, maybe going across to the other hemisphere, or maybe going across in the same hemisphere to talk to other neurons, right? So sheep have all these same connections, the same structures. And so when you're done with your dissection, you want to just label them in some way. You could also make a talking video pointing out things, or go over photos and draw over them. All right, so that's it for this dissection. But what I really want you to get out of it here is to kind of relate it to what you see in the human cadavers, which are generally much better preserved, so a lot of the features are much more distinct from that. But hopefully you can appreciate the similarities in the general organization of the nervous system after doing this dissection. All right, we'll see you next time.